Thank you. So I put together some of the experiences, lessons learned of the things that we've been doing in the past few years, and then also how they apply to the Berlin situation in particular, and also to just forming a translational center in general, as I think the Dean has very eloquently informed us in his previous speeches. The idea of BIH as forming Charité into a translational center is, of course, very relevant in this respect. So what are we talking about? Basically, I think we are undergoing or have undergone a major paradigm shift in the sense that we have begun since around the turn of the century to study each individual patient at the inside and not only the surface and the macroscopic level, but at the inside of the cells. So I've tried to symbolize that by gluing a few sequences around the magnetic resonance imaging. And I'll try to take you through a few of the things that we have done in application of such technologies and where they have provided solutions and some of the lessons that we have learned doing that. Some of it is a bit unconventional, some of it may also irk questions, and I'll be happy to answer those, of course, during or after the talk. So translation delivered, things that we have been able to achieve. I just want to start with a project that we started around 2006, 2007, when the high-throughput sequencing era began. And this applies to obstetrics and gynecology. It specifically refers to prenatal testing, and it refers to the problem of amniocentesis. A lot of the work that is being done to diagnose high-risk pregnancies leads to the amniocentesis to harvest a fetal tissue sample. The issue with that is, and it's a procedure that for certain indication is being paid for by the health insurance, and that of course then allows chromosomal analysis and further treatment steps or also parental decisions going forward. The number of procedures is quite numerous, and therefore 8% is quite a significant figure of pregnancies, or if you wish, persons lost just due to the iatrogenic procedure as a whole. And around the time that Dennis Lowe's patent also appeared, we had also developed similar technology by detecting DNA of fetal origin in the mother's bloodstream through high-throughput sequencing. The way this was initially performed basically is a process where the number of chromosomal reads is compared and statistically counted versus the average number that can be found. Up to 7% of live cells in the mother's circulation can actually be of fetal origin, and so you can see that there is a testing background. We have tried to form a company and have successfully done so by putting this assay to market very early on. So basically within a year and a half of creating this test, we have formed, together with a small sequencing company, in a group of 4 scientists, a company, and have brought forward, first of all, the high-throughput sequencing reads, which, however, is a fairly expensive procedure. So a test like that would cost approximately between 1,200 and 1,500 euros to perform. And so since the health insurance system, and I'll show that to you in a little bit, has taken some time to make a decision as to whether the non-toxic test is also being paid for instead of amniocentesis. We will have that decision in Germany only next year, 7 years after we have put in the application in 2013. We have created, the company called LifeCodex has created an assay that does Pcr sequencing by methylation. There are certain areas of the genome that are differentially methylated between mother and child, and therefore the detection of circulating fetal DNA can also be done by resequencing those particular methylated regions, and therefore the test is very inexpensive and can be afforded practically by all patients. The basic rate of the test is now around 200 euros. I'll be happy to discuss this further. Also, the question how, if you do something like this in Germany, you are often forced to sell to the international competition. By the way, we regulate these things, and I'll be happy if there are questions around this by any of you to answer that question as we go forward. A similar scenario we have addressed in gene transfer and gene editing. This is basically a scenario where in the early 90s, the idea arose, as you probably all know, that genes can be used in therapeutic intent, and vectors are being created to change the expression profile or the expressed genes or the status of expression of certain genes, or in more recent years, gene editing as a medication. And obviously, a process like that is now being used for cancer immunotherapy and various treatments for rare diseases, and needs sort of an approach at pharmacokinetics and pharmacodynamics. So basically trying to understand what a gene therapy drug is doing in the body, what's the dynamics of where the vector sits, in which cells, how many of them, and all that, and also the dynamics of the gene product and its responses as they start working in the patient's body. This is our previous scientific work. The people from the company that we have founded now call that academic history. So I'm standing next to this guy, basically on the shelf. But basically the idea is we had originally developed methodology to very sensitively read the location on where gene vectors sit when they integrate into the genome. We actually had the original intent of trying to understand the makeup of stem cell systems. So we wanted to understand how many different insertion loci are there in cells that form blood, and thereby count the activity of stem and progenitor cells. That was our original intent. What then happened, and we have been able to create a methodology and also patent that technology that does this very sensitively down to the single cell level. What then happened was that gene therapy trials came online that replaced the function of certain immune cell receptors in T cells and other types of immune cells. And lo and behold, you may recall this, in the early 2000s, some of these patients started developing leukemias. And we had been contacted by these investigators. We actually had been in contact already before, and we ran our technology across these samples and could immediately identify where the vector had gone to cause these side effects. And so that, of course, is a situation that then allows to test new vectors, test the toxicity of vectors prior to clinical application, and develop vectors along the propensity of where they tend to sit in the genome and what they tend to do for cells. So this has basically led to what is now, I think, over 200 scientific papers, because we are contributing this technology to about 60 to 70% of all active gene therapy trials that use insertional vector systems around the globe. We did, for example, the first analysis in the ADA SCID trials. We did on the first serious adverse events. We've done this for the first time in a lentivirus trial in Nathalie Cartier's and Patrick Aubour's study, and we've also provided the data for the registration of the first European gene therapy drug. Of course, the lentiviruses that are now used in cancer therapy much more widely have also been analyzed by this way, and we are in fact involved in studying some of these samples also. So this is, if you wish, a team science and a contribution of a particular translational environment that strongly enables the possibility to develop new vectors for clinical use and to try and apply such vectors for a clinical purpose. This also works for Adeno-Associated Virus. This is, again, the Glibera project and the Glibera submission to and access to market that we have been able to support by the sequencing. And so basically it's the same story like the prenatal testing. We've been able to set up with a small group of scientists by bootstrapping, so without external financial support, a company that now has 26 employees and that applies this testing and a quality management system, a validation and also accreditation, accredited lab for a number of the ongoing gene therapy trials around the world. One particular area that is of great interest, of course, more recently is the area of gene editing, where the question becomes about the on and off target effects of the new editing vectors. So how many cells do we actually manage to edit? And if these cells are being edited, where in genomic locations that are similar to the ones that are in the target region, or unexpectedly, do we also have modifications on off-target? These are assays that have been published very widely from different laboratories. In 2018, if you go back, the one that we showed for this particular application goes back to the year 2011. So we had an about 6 to 7 year advance to the rest of the field or the rest of the market in some of these areas. These technologies, of course, also are readily applicable to reorganizations or deletions or mutations in DNA for other purposes or by other means. For example, the recombination that you have in the specific immune receptors in the T and B cell system. So immune repertoire analysis is also something that we can provide. And so basically you can see that as a scientist, you can bring translation, or in fact, if you want to go beyond the frame of a single institution, probably the commercial route in many areas is the most efficient route of providing translation to a larger field. Especially then also if things like accreditation, validation, and GLP procedures have to be performed for a pharmaceutical development, because that is something that often, of course, is harder to conduct within the confines of an experimental laboratory in an academic institution. So this is also for us a mission accomplished. We have market access and contributions in all types of gene vector products. 90% of that business is outside of Germany. Of course, we don't have that many companies who do gene therapy from within Germany. Depending on the type of vector, it's a very small market. So even with this small size of a company, we can have in some areas between 20 and 70% coverage of the assays needed, or in that sense, the market. The university technology transfer concept that we were bringing this up in the 90s and the early 2000s is definitely and absolutely not supportive for any of this, because if you come to a conventional technology support area with a method patent, they will usually send you away. A lot of these organizations, and as they did to us, still dream of creating substances, creating expensive patents that then sit on a shelf and are being bought up by companies for large sums of money. In our experience, something that is a bit more procedural is also well worth the effort, but I think translational organizations, also academic organizations, need to form these spin-out companies to show at least proof of concept, if not run their own phase 1, phase 2 activities before any larger issues can be done. Again, if we have time for discussion, I'd love to talk to you about China and the possibilities or impossibilities, again, from the European side. to enter that market and what the limitations there are and what political issues we have around that. So if we have time to discuss, I'd be happy to also discuss that. In a third area that I, of course, very much like to talk about is the field of cancer, where I have spent a significant time of my career. And I think there are a few lessons that apply to all translational research in any disease that we can learn from this area. If we see the sizes of the large killer diseases, cardiovascular, neurological diseases, and cancer, we have basically a situation where about 1 in 2 of us will have such a disease in their lifetime, and about 1/4 of us at the current standing of technology will die of cancer going forward. Many of the cancers that we are actually seeing in the hospital can be cured by removal, so about 2/3 of patients who are diagnosed with cancer undergo a cure. The residual third has, of course, issues because the cancer is rare and or has metastasized or is unconventional. And that is, of course, an area with a large medical need that needs to be served. What we had tried to do in the Translational Center in Heidelberg at the NCT is think about, similar to the previous 2 systems, what can we do with regards to applying high throughput technologies. And this has been an effort that has involved very many people, from basic genetics through bioinformatics. Roland Eils has joined us here in Berlin. He's, of course, one of the heroes of this field, as well as then the clinical application. And what we had been thinking about, and as a strategy, was basically that when we were asked what does personalization in the Translational Center mean to us, we had basically said we are going to sequence everybody who comes through the door. Now, of course, that stretches it a bit far, and that's not what we ended up doing, and it's also much too hard to pay for. But what we try to do is pinpoint a patient population that has very serious issues with cancer and might best be served by such an approach. And the approach we took is that we said all the patients who are young for cancer related situations, which is under 55 years of age, and who have a catastrophic or rare cancer. Catastrophic meaning they have a metastasized disease and cannot be served by surgical removal. And so basically what we then went ahead and did with a lot of support, Stefan Fröhling and Hanno Glimm have been particularly active in this area. This, I think, also dates back to a time where Martin Kluxen, who was still at the NCT, who is here today, we took a comprehensive molecular stratification approach as a center, so running these patients through DNA sequencing, RNA sequencing, in some areas proteomic analysis, with the idea, first of all, to understand, is this feasible? Does it yield diagnostic information, and does it offer therapeutic opportunities for patients? We called this, because it is an umbrella trial that applies to all of these patients, we called this trial MASTER, not because we wanted to dominate everybody else, but we wanted to symbolize, it's basically the acronym comes from molecular aided stratification, but the name is supposed to symbolize that it sits as an umbrella over all of the other trials that would then subsequently follow to manage such patients. So here again is the indication, young patients with advanced stage cancer, patients with rare tumors. We did a fast track exome and RNA sequencing. The fast track side of it was very hard work to get a turnaround that is even clinically meaningful and not in the area of months. Required very significant work, and then also, of course, methylation profiling and other things came to that. We started in 2013, when this was still a very unpopular measure. More later in 2016, when the first X10 clusters became available, we switched to whole genome sequencing in the majority of these patients. And then this trial has also become something that was taken multi-center through the German Translational Cancer Network funded by the BMBF. So we have a situation now where there's ethics approval at all sites, combined clinical data repositories, combined tumor boards, and similar things. So for patients with rare diseases, and you can see here that there's quite a collection of them, this is an opportunity to see whether there's anything that is not known, or at least not known in this particular individual, that might be of therapeutic use, or at least might inform further research down the line. You can see here that February 2020, by now this has been a very expensive but also hugely successful operation. I'll take you through the results in a little bit. There are about 2,000 patients now enrolled in this trial in 99 different larger cancer groups and 262 smaller sub-fragments. If you look at it from the molecular side, of course, that number may be even larger. In rare cancers, there are hardly two cases that are always completely alike. A DKT master can then also be used as an informant, because the patients are consented to be able to do that, to lead to the patients into trials that try to treat certain molecular diseases. So it's basically also a very efficient way of finding patients for different types of trials across all of these organizations. And therefore, we have advanced from a situation where this was perhaps interesting and informative in about 8 or 10% of patients, to a point where we now have a recommendation that potentially could be done as a treatment in 86% of patients. Interestingly, the diagnosis that was originally made is changed in 4 to 5% of cases just by the molecular analysis, and about 11% of patients do in fact have a familial disposition, so a finding that is potentially also relevant for treating or at least observing their relatives, because they might have a similar disease. And then there are some statistical ways of testing whether the therapeutic levels can actually be achieved. What is tested and what to us has been the most interesting parameter is the question of the progression that a patient has between two different changes of therapy is longer than the one they had in the previous round of therapy. So we said this is potentially clinically successful if patients have a progression-free survival ratio compared to what they had previously that is larger than 1.3. So that is 30% longer than the previous treatment. You can see here again that there are a number on the re-diagnosis of implications that allow clinical action, or even just open any type of potential therapeutic intent as a whole. And here again is the summary of the data for the familial disposition. You can see that autosomal recessive disorders of significance and a carrier status is detected in about 1 in 20 of these patients, which of course is highly relevant for them and their families. And interestingly also, and I've put together a number of examples here, is that some of the findings that could be made are explaining observations that previously could not be put together. So for example, in Leiomyosarcoma, which is a disease of the muscle, there was the finding that a very heterogeneous mutation pattern was present in these tumors. And that of course didn't, when these were all small series, did not show any particular therapeutic approach, but taken together, the group could identify that mistakes in DNA repair, so-called BRCA-ness, so a genotype that is similar to the breast cancer gene mutation of that name is present in more than 90% of patients, which of course means that we can use therapies that are intended for so BRCA type cancers also in this group. You can see here again that this also applies to a number of neuronal tumors and other rare diseases. And I've just put together here a number of other findings. Pancreatic cancer, a certain subgroup of pancreatic cancers has been detected to have a particular type of fusion gene that can be treated by therapeutic intent. So this is a small subgroup of the otherwise very hard to treat pancreatic cancers that can be approached in this manner. Again here, neurofibromatosis schwannoma. There are certain very interesting mutations that can lead to surprisingly good results in smaller subgroups of patients. And of course, in these rare cancers, that can mean a lot to the patients who have otherwise very limited possibilities. Here again, just to quickly indicate that this is not only science for science's sake. The data is always used to run the patients across a grid of all of the available molecular studies and try to qualify them into the molecular studies and clarify the eligibility. This is with clear visibility by the patient, so the patient is informed that their data is tested in this way and is consented to recontacting. And I'm stressing that because I think that is something that we should also consider across not only cancer, but all of the activities we are having at Charité. And you can also see that, interestingly, the genetic information is very informative for qualifying patients for immunotherapy trials. This has to do with particular confirmations and also the questions how many mutations a patient's tumor has is in several indications one of the best indicators that we have that cancer immunotherapy might have a chance of succeeding. The scientific output of this then as a whole, and this is a little bit also a mission accomplished slide that I'm very proud of presenting what the group around Stefan Fröhling in particular and Hanno Grimm has done, is that we have more than 25 multi-institutional projects derived. Also the number of publications is crossing 30. I showed you about 5 or 6. We have now been able to add additional layers of patient characterization. And then, of course, this is now also reaching into the question, is this affecting other modalities and the indication for other modalities? And one particularly interesting area also is then the question, how do you now get, and I think that is also an important part of translation, how do we now get from these center, university type, large city experiences into applying such things across the entire country? And so we have been thinking about that very hard. There's one area in particular that is unfortunately in Germany still drastically underserved, which is the treatment of lung cancer. You can see that this was the historic chemotherapy pre-molecular age, median survival 1 year, and a response rate of 20 to 30% for non-small cell lung cancer. You can now see here that there are several subgroups of patients that have large response rate to inhibitory drugs, and many of them are on a succession of multiple inhibitory drugs over the course of time and can be treated with a surprising success. If you look at one of the registry trials which has the best information, and these are hospitals that are actually participating with the intent of testing patient, you can see that the testing that we have across the board in Germany is far away in certain instances from the 100% that it should be. And this is in particularly happening in smaller peripheral hospitals because the testing isn't being paid for by the health system. unless the patient is in the outpatient clinic. But if the patient is an inpatient in the hospital sits on the molecular testing bill on its own. And we try to create a situation with the help of the health insurance systems that creates a network to spread this know-how and these possibilities across Germany. So with the help of German Cancer Aid, our Cologne colleagues and myself have created what is called the National Network for Genomic Medicine, together with all of the comprehensive cancer centers of German Cancer Aid, to look after countrywide implementation of molecular testing for all of these patients. This is organized in such a way that the quality of the testing is coordinated, that the information from the testing is combined, and that a contract has been formed so that the health insurances that sign these contracts pay a flat fee for this type of molecular diagnosis, which of course seeks to jumpstart the translation of our actual findings into the hospitals across the country. So in summary, this whole precision medicine paradigm delivered the testing of the molecular makeup of individual patients coming into the hospital with whatever disease is clearly feasible. We can apply basic science technologies to do this. It complements and advances routine molecular diagnosis. It creates many therapeutic opportunities and is, of course, especially useful in situations where the diseases are rare, complex, or newly emerging. There are, of course, genetics is only the, if you wish, outer surface that was technologically feasible for the first time, but other profiling, including, of course, a view from a single cell, which I think is our particular Berlin approach at these matters, will be very important as we go forward with this in the future. So with regards to the translation delivered, a little bit of time I wanted to spend also in terms of the organization of a translational center. What is the situation that we should be seeking to do if we form a translational center, being it large or small? Our experience has been that this can be divided, I think, into at least 3 main stages. And I think you can see very well with BIH that we are probably between the first and second stage of this process. Basically, the first is the idea of forming a translational center, the principles and practice of doing so, considering that every patient that enters the organization is part of that process, and then also that every trial the organization forms is also part of this process. So this is basically about creating the interdisciplinary and combined approach of activities that one can achieve. The next step is, this first step is often only happening in the brains of parts or all of the people who are professionally participating, but it's often very hard to make it visible from the outside. I have myself underestimated the aspect of the haptic, of the perception of locations, of buildings, of the perception in the real world. The next step is an organization like this usually forms buildings and structures. You can go there. People are talking about, in this case, this was the NCT, or if you talk about the BIH, I am working at the BIH. I am working in the NCT. As a patient, I go to this structure. So basically, the second step that is relatively hard to form and takes a significant amount of time is the question of identity and visibility. And then third, but not to be forgotten, is of course then to get to that level of excellent performance and international competitiveness once you have started to outgrow the pioneering stages, which is then which programs, once you have formed that organization and that opportunity, are you running across the organization, and what are the criteria that you should be running them. if we look at the NCT in the experience and how that would reflect to the BIH, I think the formation of the visible brand, of the understanding of what one tries to do together is extremely important. And then this brand at the same time, of course, has a purpose and is filled with content. And in the cancer case, I've shown you a few of the things that we've done. This is, of course, just a small fragment of all that has been performed. But it has allowed us that we basically applied to the Ministry of Research in order to obtain funding. And you may have heard or read in the newspaper that additional NCTs will be formed around the country. So basically, once the concept is created, the mission is clear, and the competitiveness can be developed, these centers gain meaning beyond the confines of a particular organization or a particular location. And I think that's very important. In this case, our application was also considered for a second site in Dresden, Germany. And I think has been very helpful to create the notion of comprehensive centers that focus on solid tumors in Germany. And we are now, in fact, seeing the third building phase. Interestingly, in my application, I had come up with the idea, we had come up with the idea of putting an additional floor on top of the building. That proved technologically absolutely not feasible, and so they're now building the extension of the building in the front yard. So sometimes you have to have that flexibility also as you go forward. I wanted to also touch a little bit of the area of delivering translation with regards to the digital side of things, again staying in the same model. If you look at a process map of how clinical treatment is actually guided or performed, usually the patient enters the organization somewhere, he has an outpatient or inpatient contact. Oftentimes then a local decision on what needs to be done is made, the patient is sent off, and this is documented, but only in the routine system. It may happen that the patient applies to a multidisciplinary conference or even a clinical trial, in which case then later in the process, other people become involved in actually caring for them, treating them, and at that stage or exposed, documentation and data is collected. That is, of course, a less than ideal scenario from the purity of actually being able to work with the data. And of course, this process doesn't work at all for things like machine learning or artificial intelligence, where you need to collect data that you don't even know you need it, but where you are trying to do open-ended research questions. And so the design that we had thought about, and this was in 2011, for the Cancer Center is something that one would probably now call a data warehouse, where we had basically, and we are currently trying to develop a health data platform at Charité that tries to do similar things with one, I think, little difference that I will talk about it. However, basically the idea there is that every patient is going through the system considered to be a trial patient for at least their basic data collection, and also their agreement that that data can be used for research, and that data ends up intra-institutionally in that data backbone and can be accessed, of course, through a model of controlled access by those in need of the data, and by those the patient wants and has agreed to use that data. So, which is not only on the drawing, but also in real life, a much more coherent concept of processing data and arriving at these things. And so what we did was we created a warehouse that we called the there house, as a little pun on the word where, to indicate that this is where the data was. It took about 6 months to set up the system with a memory resident data warehouse. And then what happened was that a big political discussion arose between the clinical departments as to who should be able to access the data. And there was, of course, also a lot of discussion about potential pitfalls of creating transparencies around the system. So actually, to set this up technologically is much easier than the political governance of a process. Lo and behold, though, we ended up recruiting a professorship into this area. You know that if you give projects like this then into the hands of well-versed IT organizations, they create these drawings with all of these buckets and combine them into larger processes that, of course, us as laypersons are much too naive to understand. But basically, there are collection areas and then staging areas and analytical areas that can then draw data from these systems. I think it is very important that we do not forget to create this particular interface. A lot of the IT projects that I have been seeing are potential connections that IT departments and large projects can make, but there is not the possibility, or there isn't a screening tool for the physician who is working with the data on just trying out certain hypotheses. And I must say, at least from the perspective of the molecular tumor board, it would be enormously important to be able to have a lookup table within the organizations, and then, if I may dream into the future, across the entire health system, to have what you would probably call patients like me lookup situation, where you can look up the information of the last 10 cases or so that were undergoing treatment for similar issues. Of course, then also to aggregate that data and have clear statistical power is also very important, but I think we could gain a lot from the access and from the abilities of clinical researchers to very much directly access and interact with this data. One particular area that we then focused on is also the question of what happens then outside of the institution. And a project that we have been involved in the last couple of years is creating a patient data file similar to the ones now conceived by the health ministry for the health insurance system, but a bit more open in the format that allows patients to receive and transport data. We currently have a situation, especially across the German system, that patients are carrying their medical information via CD-ROM or faxes or other modes of sort of not so well suited transport. And we think that as an organization, we should be able to give our data to patients, and for our patients to be able to make decisions what happens with this data. We are doing this in all other walks of life. You all may have a banking app on your smartphone or similarly important decisions. Just for health data, we cannot do this, and so we have tried to create a patient-centered organization that really empowers the patient to control and have a holding of this data on their own. The access on the physician and on the analytical side is, of course, limited, at least in our model, to accredited, reliable sources of data processing. But there again, we think, in contrast to what the current legislation provides, the patient should have the freedom to provide data for research if they so choose. We currently have proposals for legislating patient data files that specifically should forbid data donation for certain purposes, and I think that is something that we should probably seek to change. And I think it becomes very obvious what the advantages are. I think this is a very integral part also of creating a translational center. Patients who run through our system, who have consented to donate their data for research, who have consented to participate in a trial, I think we as an organization should give them a copy of that data for their own use. And then, of course, we are approaching the brave new world of more artificial intelligence and machine learning areas. We had one particular group of this DataBox project run by Tito Sprinker, who has now spun off as his own junior research group at DKFZ, who has shown that at least for the processing of visual information, machine learning and neural networks are very well capable of supporting, or as he has written it, creating great contempt, in some ways outperforming the diagnostic capabilities of people. We will certainly, I think, see a lot of these applications, probably in the areas of image processing. We will see the more powerful applications, but again, as a system, I think we should be able to set ourselves up to process this in the space of interacting with trial center data. So before I close out with just mentioning a few of the things that we have done at BIH, I just very quickly wanted to use a couple of minutes because I have a concern that we as a society should also be adopting that paradigm. Do I still have a few minutes? Okay. Because basically many of us are of course exposed to serious or fatal diseases, and that is especially painful. This is a picture of my mother and myself a few years ago when a death of a near person or loved one would have been completely avoidable. And so I have looked at the overall discussion of what we have, what we do, for example, around cancer. We have a system that spends very little or next to nothing on prevention, but we always, when we have new forms of treatment or even are talking about prevention, we're talking about the large sums of money that cancer drugs take and what it all eats up in the system. I, in fact, looked up the statistical numbers from the Bundesamt for Statistics and found out that we are, in Germany, spending around 6 to 6.5% of our health insurances against cancer. So remember the numbers I gave you earlier. About half of us get this, about a quarter of us die from it. We are spending about 1/15 of what we're spending on health to treat it. We are spending next to nothing, probably in the per thousand range for prevention. If you want to see that as a size, this is the money we're making a year. This is the money the state takes. This is what we spend on health. This is what the public insurance spends. This is what we spend for treating cancer. This is what we spend on researching cancer. Prevention is probably a dot that is too small to be seen for more than the second row. This is what the state takes in as tobacco and liquor tax, just as a comparison for size. And what I think we as a society should be doing, we had a discussion in our prevention work against colon cancer. Where are other areas where our society has been very successful in addressing fatal dangers that are happening in complex systems? We have this in traffic, we have this in the workplace, and we have this in aviation. No plane crashes. Our traffic accident numbers were peaking out at the 70s at 22,000 people a year. We're now, although we're driving 4 times as much, we're down to less than 1/10 of that. How have we done that in those systems? There is a concept coming from Scandinavia that has been adopted in Europe around the 80s and in Germany around the 90s, which is called Vision Zero. And the very clear statement on a Vision Zero is basically that we find no avoidable death acceptable. We are spending enormous amounts on single cases of prevention, and we are reanalyzing every single case of an accident, of a flaw in the system, and addressing it by optimization. And if you recall this, over the course of about 30 years, we've been able to drive this number down. And I'm very convinced, I'm running out of time a little bit because I enjoy talking about this so much, but we need to be focusing our resources on actually going into prevention and intercepting disease early. And it's actually, as has been in these other systems, it's hugely cost effective because the true cost of losing a life, even in economic terms, is very substantial. And a zero can be achieved. You may have read in the newspaper that Helsinki and what was the other one, Oslo, had 0 bicyclist traffic fatalities in 2019, 0. If you think of the Berlin situation, it's a bit different. When you look at 22,000 deaths, which was the peak number of traffic fatalities that we had, is what we are losing every year for colon cancer. And if you see what we could be doing, like our Dutch friends across the border, for example, are doing, is sending a prevention test to everybody in the country over 50. We are clearly coming up short, and I think this is also a question of translation and of developing disease prevention and interception for us as a center and the political work that we need to be doing. I'm almost out of time. I briefly wanted to touch the BIH, but probably you have heard a lot about it. So I will just focus on not on the structures that have been built, but the particular area that I am working together with especially Alexander Kranich and Zane Schmidt, which is the clinical study center that tries to form a unified system of supporting clinical trials in the Berlin environment. We want to take this forward into a defined and controlled process to optimize the support for the clinical investigators to run innovative clinical trials like the ones I've showed you earlier. We would like to create and enhance the transparency around this process. There's also an issue of quality control and governance that we as an organization need to be taking care of. We have created a new outline of all of the services, and more importantly for you as a potential customers, we are creating a one-stop shop of a single point of contact and entry that allows you to contact this organization if you have interest in this. I think, and we are currently having that discussion internally, we should be also creating, similarly to what I showed you about MASTER, an umbrella trial that consents patients for donating their data and being recontacted once they enter our system. And we are creating a number of service and innovation structures to perform this, as well as together with the Charité leadership, the area of quality management of that entire process. There has been a long shopping list of very beautiful work and achievements that we had to come up with in the first half year to combine the existing organizations in a unified platform. I think we're well on our way of doing that. There's also very significant service to more than 200 and in some areas more than 300 ongoing studies already. I think this is also something that you are going to see a lot more of in the next months as we are advancing this. There's also a governance framework and information material being prepared that will allow you to gain an oversight of what we're trying to do. And I have arrived at my summary slide. I would quickly wanted to take you back still. We have been able to deliver aspects of translation through the testing that we have provided in obstetrics, in gene transfer, in gene editing, in cancer, so that precision medicine approach of studying the patients deeply and early, I think is useful in very many different areas. I think there are certain design features of a center in terms of how it's set up as a whole, how we are setting up the processing of the digital age, and then I hope I've been able to convince you also as a society that we can take forward as an organization to succeed in what we're trying to do. And with that, I would like to thank you.
Yeah, thank you very much. The paper is open for discussion. Are there questions?
Nice to see that there's a, let's call it, brain gain from the south of Germany, from Heidelberg. I have two questions in the first part of your talk. So one was on the, you call it detection of DNA in the origin. To my knowledge, it's mainly on triplication, 21, 13, and 18, and then you can also differentiate X, Y, X, X.
Yes.
Is it also possible, I think the trick is to have a very good antibody, right, to suck out the DNA, or is this done technically, and then the question would be, is it possible to also check for mutations? So there are some certain diseases now, Cheng and Thumgap, which are so common. Is this possible? That would be my first question. And the second is to the gene therapy, and you're also the stuff you mentioned on cancer sarcoma. You mentioned lentiviruses. I'm really naive there. Are people not using AAVs for gene therapy?
So to the first one, the resequencing for particular, if you are looking for particular mutations, you can do resequencing, but you are not getting a whole genome equivalent from the circulating DNA. This is a statistical process on where you're basically looking in the first type of the methodology, how many reads that would locate to chromosome 21, for example, or 15 or 18, can you find compared to the average of the reads and related to the size of the chromosome. So it's a statistical process. It doesn't have anything to do with antibodies in that sense.
But to get the fetal DNA out of
no, that's basically you're prepping all of the DNA, and you're doing in the first one, you're doing it by weighted analysis. So basically, do you have more reads of 21 than of the other chromosomes? And on the second one, yes, it is a targeted resequencing of certain areas that mother and child share, but we're looking at what is the degree of methylation in one versus the other. Because the children in some areas are hypermethylated, so the genes are silenced and the mother is active in these particular locations. And so there you can differentiate. With regards to the vectors, yes, of course, there are AAVs and lentivirus vectors depending on purpose. The AAVs are only partially integrating and make a very nice expression, especially in liver cells and cells that are post-mitotic, because they form their own sort of DNA and do not need that cell cycle integration process as much. The lentiviruses are much better for modification of cells that have a very high turnover, like blood-forming stem cells and T cells, as they proliferate.
Maybe I may add another question. In the beginning you said that researchers can and have to become business people, more or less. So do you think that's really the best way to go? Because some people might be very good or interested in research, but not so much interested in really doing the business. They might be interested in supporting it and pushing it, but so don't you think we rather need teams to do it?
Yes, to all of these questions. So you see me standing here.
Yeah.
So I've started to establish these companies. I'm not a business person myself, but I had a very clear understanding of the tasks that needed to be performed. And we teamed up with other people. Also, we recruited, for example, for Life Codex immediately, a person who was very well versed in running a biotech company and is not a scientist himself, But then he turned out to become a mixture between a business person and a scientist. He was very instrumental in developing that second stage assay. But what we are currently doing in the university system is that we're keeping it in the academic side for too long and running demonstration project after demonstration projects where the cost of these demonstration projects would already be enough to found a small company and try and go commercial early in some of these instances, especially in the diagnostic side. If you look at the American organizations, they do this. The great inventors and scientists are founders in multiple companies, and they're not business people, but the going out into forming a company is much earlier than we do this here. Technology transfer here is always a bit about, you know, we shouldn't, not yet, shouldn't we do another demo project? And I think we should be more aggressive. I think I tried to show an example. Prevention is hard.
Is it really harder?
Well, in a way, yes, and in a way, no. Of course, so for example, for colon cancer, we had a stool test for blood that wasn't particularly good. We offered a colonoscopy, but the take-up rate for that is a bit lower because many people do not like the idea. In more recent years, we had an immunological blood test that tests for the presence of human blood. And that's much more informative, and that's the one that the Dutch use. And so that is a filter. It doesn't find all of the patients, but it clearly very much enriches the one who have a problem. So it's neither leak-proof, nor is it very sensitive. So the sensitivity and the specificity are not very high, but if what you can do at a population scale is actually quite good, but that is only available in recent years. In other areas, we had big discussions. For example, PSA testing for men has led to an overtreatment, right? So we have too many resections, and we are still trying to study which of the patients need an aggressive approach and which can we wait. So yes, you're right, we're not spending enough on it, but it's not an easy topic.
That you don't get cancer.
Yes.
What is the reason to get it, and what can we do to prevent
yes.
it really?
Yeah, beyond the things grandma already knew, right? The smoking and the heavy drinking, if we leave that out, that would cut it down to about a half. And then there are the...
But also to bring this into the community would,
yes.
I think, give more output or decrease maybe
yes.
cancer.
The motto for our Vision Zero campaign this year is going to be that we wanted to talk about the price of cigarettes. German cigarettes are the cheapest in Europe. If we add €1 on a pack of cigarettes, it would offer us 3.7 billion euros that could be used for anything, for example, prevention.
And it would be preventive in itself.
Yes.
Because probably we won't get the 7 billion, but only 5 billion, because some people would
yes.
stop buying cigarettes.
Especially the school kids are very price sensitive, as we know from around the world.
Are there other questions? Yeah.
So in one of your slides, you had a relatively low N number. I think it was a sarcoma, 49 patients.
Yes.
Takes it as a hidden critique, but then there was a kind of diversity within your data set. Are you also using deep learning or neural networks, not in neurosciences, but in computer sciences, to figure out structures? So if you have a limited N number,
this is something that is, of course, going on in the basic science of also genetic analysis, yes. In this particular project, we're not there that we're saying we're doing it from the clinical side, but some of the pipeline development of interpreting molecular changes in the comparison, because these are always comparisons between the normal genome of a person, so we're always doing the normal tissues, and then we're comparing it with the tumor tissue, and are basically doing, if you wish, a subtraction analysis, and that is, of course, something where these types of processes are being looked at. There was a paper in Nature, I think about a half a year ago, that showed that at least it could be a lot faster that way, if not also more sensitive.
Okay, then I don't see any further questions. We thank you.